Okay. So hi, hello everyone again. Hello and welcome. Microbehunter here. Yeah, first of all, I some apologies for the slightly confusing starting time today. I actually wanted to move the start of the live stream of 15 minutes later today because I wasn't sure whether I'm going to make it back home in time, but I did make it back in time. So I basically I'm starting now. Okay. I'll basically it was just a little bit that I've had some personal time constraints, and that's why I thought I'm going to move it 15 minutes later, but I didn't make it in time. Yeah, so what I'm going to do is I'm just going to start now because I've seen that several of you have already joined in. We're going to just take it from here, and I hope that others basically don't miss too much of the start. Okay. Today, I would like to continue a little bit the thing that I started last week, which is the whole issue about slime molds. I've been spending the last week growing slime molds, more or less successfully, and I would like to simply report back a little bit on my success and not so successful experiments. Okay, so we're gonna do a little bit of stereo microscopy today as well, and maybe, of course, also a little bit of compound microscopy. And I would like to explain to you a little bit of what I've been doing the last couple of days, because what you can already see here, well, other side over there, that is actually an agar plate with a slime mold, and I would like to talk about this a little bit more today, because I actually said that I'm gonna start 15 minutes later, and some of you might now expect the starting time to be a little bit later, but I see that many of you already started to join in. I'm just going to go through everyone again here, as like in every week. Hello from the UK, from Pennsylvania, the United States, Netherlands, Colorado, from the UK, from Netherlands, from Australia, from Poland. I'm fast. Slovakia, from Bodensee. Okay, that is basically in Central Europe again. Northern Germany, from London, United States again, Germany, Copenhagen, Denmark. Okay, so hello everyone. A little bit some info if you're basically new to this format. After approximately 1 hour, I'm going to answer again some questions. If you have any questions, please feel free to type them in the live chat. But off-topic questions I would like to answer approximately a little bit later. Okay? Yeah, from Vienna, so not so far away from where I am, and also from New Mexico. So I'm going to dive right in to explain to you a little bit of what you actually see here on the side. Okay. So first of all, here at the top, that is the view of the stage of the stereo microscope. And what you see over here, this here is a cereal grain, oats, because slime molds like to eat oats. And what you see here, this yellow network here, that's the slime mold. And I started basically last week with that, but in the meantime, I was able to grow it quite a bit. And basically, I would like to show you a little bit what happened. I've been feeding it. I was basically feeding it this oat here. So this oat here was covered with this yellow-orange slime mold. And then all of a sudden, the slime mold started to spread out in a network like this, looking for new food. So it likes doing that. Yeah. So it first starts to digest the food that I've given it, and then it likes to spread out. Okay. One of the reasons why it does that is because the slime mold starts to build up waste products, and it's trying to escape now from those waste products. So I think it's interesting because it did not really finish digesting it, and it's already starting to spread out. Yeah? I'm just going to show you also some of my other plates that I have on the desk here. So that's the desk view. And let's move everything away here. I've got a couple of other plates here. I'm going to move this down now. Look, let's remove the cover here. You see, it's basically I put some oats here in the center, and you see that now the slime mold has covered the oats here. So I'm going to expect it to spread out soon. The plate here is basically agar. So what I've done is I've used agar. I just bought this. It's this powder here. It's actually used for making food. So it's not even laboratory grade. Yeah, it's a powder. Yeah, and what I've done is I used 2 grams of this here, and I mixed it with 100 milliliters of water. So basically it's a 2%, and then I microwaved it. That's all I did. I did not properly sterilize it. I just microwaved it, and then I poured those plates. And when they basically cool out, then you get something, a gel-like solid thing here. If it starts to dry out, and it will start to dry out, then all you do is you add a little bit of water here on the side, and it's going to be soaked in, and then it's not going to dry out anymore, right? So that's basically something new compared to last week. And from last week, you probably already remember that I've made some filter paper here, and it also works. However, I think with the agar it looks a little bit nicer. So we're going to put this also under the microscope. But what you see over here is that you look carefully. You zoom in here. You can see that it's already starting to move a little bit on the filter paper here on the side. So it's trying to escape as well. So we can take a small sample here and put it also under the microscope, because this was something that last week we were not able to see a lot. Yeah, so I've been basically growing it. Then also over here on a small version, you see here as well. And I've been trying different things here as well. And this one didn't work quite as well yet. So you see that there are different ways of growing them. And this one seems to be quite promising. So what I'm going to do, actually, it's kind of my intention is to take this away, put it on filter paper, and then watch it spread. And then when you dry the filter paper, then you actually are able to preserve it this way. Yeah. So what I'm going to therefore do is I'm going to also show you a time-lapse video, which is really cool. But before I do that, I'm quickly going to have a look here again. Yeah. So did the slime mold last week finish his meal? Kind of. Because what it does is it will start to spread even before it finishes completely digesting it, because it wants to escape from the waste products that it made, right? I don't know what these waste products are. Maybe it's pH sensitive. So usually when wastes are made, then there is it's acidic. So it's a hypothesis. So I think it might actually be pH sensitive, but that might be something that I can test in the future. Yeah. So greetings from the UK. Okay. I'm just going to skip ahead to comments that are directed to me. Fun fact: microwaving was invented for sterilization purposes. That's it. It's interesting. So normally you have to autoclave it using a pressure cooker. Yeah. Hi guys, a little late today. What did I miss? You did not miss very much. I was just saying hello. I wanted to start the stream. For those of you just joined in, I actually wanted to start the stream today 15 minutes later because I just returned home. But I actually did make it a little bit on time, so I decided to start the stream right away. So I've been taking my time a little bit here right now at the beginning. So this is kind of the thing here. I also mentioned in the description or in the title that I want to do a little bit of staining as well. I do want to test something, and I'm not quite sure whether it works, but I would like to try to stain the how do you call this, the slime mold. Before I do that, okay, that's the wrong one. Before I do that, I would like to show you a 5-minute time-lapse video that I made over the last couple of days. Simply to show you how the slime mold is growing, because in time-lapse you're able to see something that you're not able to see. So the time-lapse is approximately 30 times faster than in real life. So it's about 5 minutes long. I'll be commenting this here as well. And I'm just going to start the time-lapse because I think it's really nice. Okay, so look at this. This is basically the slime mold on an agar plate growing. Yeah. And you can actually see how this under my stereo microscope. Yeah, I think it's pretty cool. And you see that actually it's kind of pulsating a little bit. It's not growing or flowing constantly, but it's a little bit pulsating. And the reason is because in the slime mold, it's sending off chemicals. Yeah. By the way, this here in the thing at the top, the yellow thing, that blob in the top, that is an oat. yeah, which is still food, which is still covered, and you can actually see now how it's spreading and how it's pulsating. And the reason why it's pulsating is because there are certain chemicals released that allow the slime mold, the parts of the slime mold, to communicate with each other. I don't want to say individual cells here, because a slime mold, what you see over here, actually is not made of several cells. It's actually the plasmodium stage is actually like one gigantic cell containing thousands of nuclei. So that's kind of, I think this is pretty cool. I don't know what you think. I mean, look how it's pulsating. Yeah, it's about 30 times faster than in real life. Yeah. The question is this the mycelium growing? We do not call it a mycelium because, strictly speaking, it is not a mold. That's a little bit. They're called slime molds, but they're actually myxomycetes, it's called, and they're not molds. They're amoeba that kind of fuse together and communicate with each other using chemical signals. Yeah. So so that's kind of a very unique living thing, and I think it's fascinating because when we put it under the microscope, we're not able to see individual cells because actually all of the cells have kind of fused together. These are not separate cells anymore in this stage. Yeah. So this is really is this a syncytium? Yes, basically that's the case. Yeah. So they call this the so-called the plasmodium stage, and when you put it under the microscope, it's kind of like an irregular mass of things. So what you see over here now, this is the white thing is a rice grain, and I put the rice grain there, and look what happens because those. It's gonna flow back. It's gonna float the other way. It's gonna go to the rice grain. That's kind of fun actually to see that. Let me. Here's the arrow, that's the rice grain, yeah? And look how this here is now basically retreating and it's flowing back and it's trying to now cover the rice grain, yeah? So first it was trying to spread out, then it kind of discovered there's food there, and it was kind of flowing back, and now you can see that it's kind of slowly starting to cover the rice grain. I did find out that it doesn't like to eat rice very much. The reason is because this is a raw rice grain, and I think it might be a little bit too hard. So with boiled rice, I think it works a little bit better. But essentially, it tried to cover it, and then it tried to spread out again because it had, I think, problems digesting it. Yeah, that's another one over here. Look, and here you can actually see this pulsating movement much better. Yeah, it's actually pretty creepy if you ask me. Yeah. Does the mold have many more organelles too? Well, it is a eukaryote, so it must have definitely mitochondria and other organelles as well. Yeah, because it's a eukaryote, but you do not see individual cells. This is a tiny blob that I used. I transferred that with a tiny toothpick. Yeah, and even that blob it's already pulsating, and it's kind of really weird. Yeah, where is the slime mold found? It's one of the questions. This particular one I bought. So basically, they send it to you on a piece of paper. You put the paper on a wet filter paper, and then it starts growing. But they can be found actually in forests, and in nature. They like it moist, and they don't like too much light. Yeah, but those slime molds are actually sold also for kits sometimes with equipment for students, for example, in schools. So, but this particular one I bought for a couple of euros, and you can even find them on Amazon. Yeah, they'll. So, and I'm just checking again. I wonder why it's pulsating that way. It's pulsating because every time when it releases certain chemicals to communicate, then the wave of chemicals actually causes this pulsating movement. Yeah. So you see over here that this pulsation slows down a little bit. No, yeah. It seems to be self-organizing. It is self-organizing. Yeah. So what they have actually done is, if you go online, they have actually placed food particles, over the petri dish, and it kind of connected those food particles almost like in a subway network. Yeah. Can slime mold dehydrate and survive winter in that way, or does it depend on spores for that? No. Yes, it can survive, but it does not, and it is also able to form spores, but it's also able to survive without spores. So what you have is, and I should have actually prepared that. Just a second. Yeah, here I am again. For those of you who were not did not see this last week, I'm just going to show you. And then, yeah, the desk, here it is again. It came in a little, but basically that's how I ordered it, in an envelope, and it came in an envelope in a little plastic bag. Yeah, and there were pieces of paper in here, and that is the dry slime mold. And if you put this paper now on a wet filter paper, then it's going to start growing, and then you have to put some food next to it, and then it's going to start eating the food. Right, so it is able to survive even with. It is able to form spores, but it's also able to survive without spores. And this is the dry form is called the sclerotium, and yeah, and that's what I'm also going to try to make here. Yeah, so so yes, it is able to yeah. So Joe said, "Well, what's this? Sorry for a bit late today. Well, it's a slime mold." Okay, so it's kind of a follow-up video of last week. Yeah. Okay. So let's have a look. What I'm going to do now is I'm going to go back to the stereo view here. Where is this? Here we go. I'm going to remove the arrow again. Okay, but just going to show you a couple of other parts here. Yeah. On the you see, this here. Is some oat. And why is there no slime mold? Because the slime mold already moved away. Yeah, it's pretty disgusting stuff here as well. This is the rice grain. The slime mold is not here anymore. Decided nothing interesting here as well. And now it's over here. Yeah, and what I could theoretically do, and I mean, actually, I wanted, yeah, I could try that. For those of you who were could not see that last time, I'm looking for my. Where did I put it? For the oats, just a second. I've got such a mess here on my table that I don't even know where I put it. You know what? I could take some rice here. Just a second. I prepared a whole. It's kind of interesting. I prepared a whole jar of food, and okay, well, I'll just use this here then. You know what I'm going to do is I'm going to take this here, the desk view. What I'm going to do now is I'm just going to take one of those oats here, one of those fresh oats over here, right? And I'm going to put it now on the next to the slime mold here. Yes, where is it? Here we go. See, it's already slowly starting to grow here on yeah. And let's see what happens. I'm going to now quickly move over, so I'm just going to disappear. Just check what I'm doing over there. Okay, here we go. And then actually what we should be able to see now is we should be able to see that over the next couple of minutes or so that this food is going to be covered hopefully with a slime mold. Okay. So, what I would like to do as well is I would like to take a little bit of the slime mold and put it under the microscope. Under my compound microscope, because this is actually something that I had problems last time because I simply did not have enough of it. So I'm going to take some off here. I'm going to read now occasionally also some of the questions here. Can it dehydrate? Is it possible to use methylene blue for quick staining bacteria just by applying it on a fixed by flame specimen instead of a more complex procedure? No, Löffler. Generally, yes, it should be possible. I'll talk about this a little bit later, okay? Is it smell bad? Does it smell bad? I will tell you the following, that interestingly, those that and I don't know why really, those with the filter paper, yeah, actually smells a little bit bad, but those with, so maybe there's some bacteria growing here as well, but those with the agar plate smells completely neutral. It's really interesting, yeah. So yeah, so both is possible, but yeah. So I'm going to try the following now. I would like to put a little bit of this here, this slime mold, under yeah, on a microscope slide, and let's have a look at it under the compound microscope. Why? Because yeah, it's interesting, and because you're going to see that it's really not. Yeah, it's a very irregular mass of something. Yeah, it's not something defined. So it really, normally we would expect some individual cells, but over here we don't have individual cells anymore. So I somehow have to kind of scratch off a little bit of this material, of this slime mold. Okay, it's difficult to get off. So here it is, and so that's enough. I'm going to add a bit of water, and here, look, here it is. I put it next to my monitor. These are the oats that I was looking for just a minute ago. Yeah, had it hiding right next to my monitor. So that's basically the slime mold food, just regular oat. I can show it to you here. Yeah, as well. Yeah, this was the thing that I was just looking for. So let's take a little bit of water as well. Just a second, I need a beaker. Here we go, a little bit of water. Because every time when you make a slide mount, you have to add a little bit of water. Here we go. And yeah, I'm just going to drop a cover glass directly on it. And maybe we can also do a little bit of staining as well. You see, it's not, yeah. This is how it looks like. And don't be disappointed, okay? It does not look particularly interesting, right? But I just, yeah, want to show it to you for the sake of having a look at it. So let's go here. Yeah, so don't have too high expectations of it. So let's turn it up with those lines that you see might be some of the fibers from the tissue paper. Okay, so but let's go up. Yeah, so those lines that you probably, because I scratched off some of the fibers from the filter paper that it was growing on. So the lines there, those are not part of the slime mold. Yeah. So we just ignore that. This is basically how it looks like. It's a completely, yeah, undefined mass of something. I wouldn't even say of cells, because it's actually, strictly speaking, it's not made of separate cells, because all the cells have fused together. And somewhere in here there must be thousands of different nuclei, and all of the cell organelles must be in here. But yeah, as you can see, no individual cells visible, just as expected. Yeah, we can go up yet further a little bit with the magnification. Yeah. So, yeah, you see, yeah, very, how do you say, yeah, amorphous mass of something. Yeah. And yeah, this kind of also shows a little bit that the slime mold, yeah, indeed, there does not have individually visible cells here. Maybe there is some interesting movement going on here. By the way, I do also have to correct a mistake that I made in the last video, because somebody asked me, why does it move? And I said, well, these are microtubules that make the movement. But that's, strictly speaking, not correct. These are so-called actin and myosin filaments. These are also protein fibers. So now the movement that you see here, yeah, it's also quite interesting here. So maybe that is some of the movement of the slime mold as well. Yeah, but it's yeah. But there are protein fibers inside the slime mold that slide past each other that essentially allow it to move. I'm going to interrupt myself again. I'm just going to I'm going to skip over the off-topic questions. I'm going to talk about them later. Yeah. The agar dish might be more sterile than the paper one and therefore have less bacteria. That is possible. The point that I just also want to make is that with the agar dish, the agar plates that I made, I did not add any nutrient. Normally you add some kind of a yeast extract or some nutrient medium. I did not do that for the agar plate. Okay, so this means that normally you would have a whole bunch of colonies growing on here. That's not the case because it's just regular water plus agar, microwaved it, and that's it. Yeah. So the only food that it gets over the oats. Yeah. So there is quite a bit of active movement happening here right now. Yeah. So and this is basically these cells are moving on their own. Yes. So are slime molds unicellular or multicellular? You see, that's the interesting one, because slime molds, and that's why they're studied, are a unique, how do you say, intermediate form between being multicellular and unicellular. Because the important thing is an organism that is multicellular is not only defined by having many cells, but the cells have to be also specialized. Yeah, so that's a very important distinction. If you have a bunch of cells that are all the same, then essentially, yeah, it's still not, per definition, not multicellular. But in the slime mold, what they do is when the conditions are right, they will form spores, and they will form structure sporangia that form the spores, and there seems to be already different types of cells involved here. So those slime molds are an interesting thing where essentially it's already starting to go a little bit multicellular, but actually, yeah, not to the extent that we have it, of course, in higher organisms. Yeah, so that's why they're studied, because it's this interesting transition zone. Because under certain circumstances, it's able to form those spores, and that's a form of cell specialization that's already going into the direction of being multicellular, right? So essentially, if you ask me, is it unicellular or multicellular, then I would say it shows characteristics of both. Yeah. Looking at bright field, no, I'm looking at DIC. You want to see it in bright field, then we do something like this. I have to turn it. That is now bright field. Yeah, that's now bright field. Yeah. So is anything more visible with a Reinberg filter? I wouldn't say more visible. I don't have Reinberg here. Right, unfortunately, but what I could do is I could try phase contrast, just for because it's kind of interesting. This is now phase contrast. Okay, yeah. So you see that it just looks a little bit different here. Yeah, yeah. Do you keep the plates with the slime mold in a dark place? The thing is the following, you should keep it in a dark place, but I didn't, okay? Those slime molds generally like to have it darker, and I don't put them into direct sunlight, but I did not cover them either. But if you want to actually make those sclerotia, the dry form, you should keep it in a dark place, I read, because this promotes the growth, yeah? But I've not kept it in a dark place and it still kind of worked, yeah? The actin and myosin works like the ones in the human, yes. Yeah, these are basically fibers that are sliding past each other. In our case, it's a muscle movement, and in the case of the slime mold, it's the movement of the slime mold itself. Yeah? Let's have a look here. I don't know. I hope that this is already starting to show. No, yeah, almost. Yeah, let's wait a little bit. Yeah, and let's see whether it's going to take over. So another thing that I want to try a little bit today, and I don't know if, maybe I'm going to try that a little bit later. I'm not quite sure if it's successful or not, but it's been in the instruction manual that came along with this slime mold. It said you should try to stain it, and if you add a little bit of stain, then what's going to happen is that you should be able to see the stain traveling along the slime mold, right? This is what it was kind of recommended that we try. I would like to try that, but only after I quickly showed you how this is kind of in real time, how it's kind of doing this first, and maybe I'm going to add a little bit of crystal violet stain. Yes, I know that over the last couple of months, years, I've always been working with methylene blue called Löffler's solution. I have it over here somewhere. Look, let me show it to you. But I recently discovered crystal violet, and that crystal violet actually has an interesting advantage. I would like to share with you. Just a second, the desk view. So I've got here I mixed myself a couple of stains. So that is ink, fountain pen ink. I've got over here, that is acetocarmine. That's a red one, a red dye that I mixed. Iodine, that's just the iodine solution, which we all know already. This is methylene blue, that's Löffler's solution, right? And I've been using that one. However, there is a small disadvantage with that, and that is that this one contains alcohol as well. Potassium hydroxide, KOH, and alcohol. And this basically means that it kills off the whatever you're staining. I mean, it's the whole point of it, right? But therefore, it's not suitable for live staining. And I was a little bit surprised that the crystal violet that I have over here, which I just recently obtained, this is a very, very strong pigment, a very strong stain. But to my surprise, it did not kill the nematode worms that I was staining. Yeah? The other stains actually like to do that, and so I was thinking maybe if I use a little bit of this crystal violet, maybe it's also we're able to do maybe a live stain as well. Okay, and so this is why I would like to add this here then later a little bit and see what happens. Yeah. What's your opinion on the digital microscopes? I will talk about this later. Okay. Violet. Okay. Yes. So there are some comments here. So let's have a look here again at the stereoscope. It takes a little bit of time always for it to kind of switch over, and maybe I don't know. I'm just looking, and I could actually try this as well. So just that you see how I'm doing that, and because I have a plate, and this might not be enough time to do that, but. Look, this is a fresh agar plate, okay? And yeah, it's already a little bit thin here, so this already means it's kind of starting to dry off. And I'm going to simply take a small amount from here, and I'm just going to transfer it over. And I'm going to put it on here. Okay, and interestingly, what happens is that I found out that here on this agar plate, it likes to spread also fairly quickly. And I think, again, the reason is that it tries to escape the waste products that it makes. So it kind of knows where it's already been by kind of sensing the wastes on the surface, right? Because right now it's digesting it, producing probably acids or other wastes, and therefore it quickly spreads to find new places for food, right? And this is also one of the reasons why it kind of escapes fairly quickly, even before the food has been fully digested. Yeah? But that's usually how I've been kind of reproducing those. And let's check again the stereo view over here. Okay. Yeah, we're going to see how this usually takes some time. So for those of you who joined in a little bit later, I'm going to go back again and I'm going to quickly show you again the video, the time-lapse video that I made, which I showed at the beginning here. Yeah, so this actually shows how the slime mold is spreading. Yeah. Yeah, this is now also on this agar. I think it's pretty fascinating. Yeah, and one of the things that I can do now, look, I can actually speed it up a little bit. Yeah, here as well. Yeah, yeah, and it has this interesting pulsating movement, and with every pulse, it moves forward. The total time of these videos is around 1 hour. So this, is I've been speeding it up quite a bit, but the movement is I wouldn't say very slow, but it's in order to see the pulsating movement, you have to kind of speed it up. I think this is one of my favorites one here. Yeah. kind of inching forward a little bit. Yeah. I think the slime molds would excrete their waste outside. Yes, of course. Like cells, what they do is they produce waste, whatever, right? And they are accumulating, and therefore there is a concentration gradient of wastes, and therefore they know essentially into which direction they have to move, yeah, to explore new areas. So what actually happened in one of my plates a couple of days ago is that the slime mold actually tried to crawl up the glass wall of the slide, yeah? Which basically was kind of surprising to me that it actually tried to do that. Yeah? So these things have a really almost, some people say almost like a form of intelligence here. Yeah. What's my recipe for the agar base for the slime mold? Yes, quite easy and straightforward. The recipe is you make a 2% agar. This means you take 2 grams of the agar, and basically that's the one that I just bought for cooking. Yeah, agar, 2 grams plus 100 milliliters of water. I even used tap water. I did not use distilled water. And I microwaved it. I properly boiled it until it was all of the agar was completely dissolved. And then I poured it into those plates, and then I waited maybe an hour, 2 hours until it's cooled out, and then it was solid. Huh? So it's pretty easy. Making those plates actually was only, I don't know, 5 minutes. Huh? I just added 2 grams of agar, 100 milliliters of water. It's boiling, bubbling, so be careful that it doesn't boil over, right? So you might want to make sure that the glass is sufficiently tall. And I was boiling it maybe for half a minute until everything was properly dissolved. Allowed it to cool down a little bit, otherwise you're going to burn yourself. And then I poured the plates, and that's it. Yeah, finished in 5 minutes. Yeah. Off topic, kind of send you a link to an amoeba time lapse. Oh, that I did. Okay, thank you very much. Okay, thank you. Okay, so this is great. I don't have currently, because I'm doing the live stream, I don't have access right now to my email, which is on my other computer. Yeah, I'll try to show this. Yeah, so it looks as if only a few regions are actually moving, growing. Only the tips of the branches, the stem is not surrounded. Yes, so that is an interesting observation. As a matter of fact, when you look at something like this, where is this? Yeah, this one is a good one. If you look actually like this, there is the growth indeed seems to be mostly only on the how do you call this, on the sides here, right? But actually there is a lot of stuff flowing here on the branches as well. Yeah, so that's but it's probably not as visible. Yeah, so what happens is that as it explores new areas, the slime mold is kind of flowing forward, and when I place a food over here somewhere, then it's kind of flowing back again, and it's covering the food again. Yeah. So it appears like it's growing only on the tips, but actually it's kind of the rest, all of the other branches are kind of supplying, the whole thing. Yeah. So really, I don't know. I like these guys somehow. So I don't know if let me quickly have a look if this takes a long time for the I don't know. You know what I'm gonna do? I mean, you've seen it already. I do want to try it, and I'm not quite sure whether maybe it's gonna kill the slime mold or not. What I'm gonna do is I'm just going to. We've seen how the slime mold moves anyway, and I would like to try the following. Now, I would like to try to put a little bit of this crystal violet stain on here. I have not tried this before. Okay, I don't know how well it works. The reason why I want to do that is because this was actually recommended in the instructions. Yeah, so I'm just going to take a little bit of this crystal violet into my pipette. Okay, which you didn't see right now. Okay, so there's a little bit of this blue stain here on the pipette, and I will try to drop it now a little bit on the how do you say, on the oat there, and I kind of hope that we're able to see how this crystal violet actually is spreading along. I have not tried it, so if it doesn't work, it doesn't work, right? So just a second, watch what I'm doing. So that's the how do you say the thing that we should have tried. And then I don't know if it's it said, the instructions said that you should be able to see how the stain kind of travels along, and then you should be actually able to see the movement. I really hope that the crystal violet doesn't totally destroy the cells here. I don't know. We're gonna see. Okay, so we're gonna be a little bit patient, and with a little bit of luck, it's gonna work. If it doesn't work, we've also learned something, yeah, new here. Yeah. Did you mix phenol to the crystal violet stain too? I did not. I will be honest with you. I did not mix anything together. This was a ready-made mixture that I got from the lab. Okay, so it could be, if there is phenol included in there, then it's probably gonna kill off whatever it gets in contact with. However, I did try to mix this crystal violet with some nematode worms. I can try this as well, just for the fun of it, and they actually survived it quite well. Right. I've also got some water fleas here that I would like to feed a little bit, and that's also something I would like to basically do in the second part of this video, to try to feed the water fleas with stained yeast cells, yeah, and to see if we were able to see them being eaten up a little bit better, yeah. Just a reminder, you said you'll talk about your opinion on digital microscopes. Yes, I will do that. I will usually always answer the off-topic questions in the last half an hour of this video. So I'll go through all of those off-topic questions later on. So I don't know. We're gonna see how well this actually works or not. Okay, but let's do the following. I would like to slowly move on, and we're gonna be patient with that, because I would like to, I would like to do a little bit of going off to the next topic. I would like to try a little bit of yeast staining as well. The reason being that I would like to feed the yeast, the stained yeast, to my water fleas, in the hope that we're able to see them being eaten a little bit better. I've not tried that yet. Okay, so this is also going to be a little bit of a new how do you say, experiment. And those of you who have joined in already in the past know that I like to do a little bit of, how do you say this, trial and error and experimentation. So I'm going to give you the desk view. You see already it's blue. I've already done a little bit of staining here. Yeah. And I'm going to, where is this here? I've got some, I got myself a spatula. So this is a little bit of, add a little more water here. Yeah. So this is some yeast. I'm just going to suspend it a little bit here, because that's going to be the food for the water fleas, for the Daphnia. But I would like to add a little bit of stain here as well, simply to make it better visible. So actually has been also a recommendation in one of the microscopy books that you should stain the yeast cells before feeding them, because it's simply easier to see then how they go through the digestive system. And if we don't use too much of this stain, then it should not harm the organisms either. So I'm going to wait a little bit for the yeast to have completely dissolved. Okay. And I'm going to now prepare some of the water fleas. Daphnia, they're here. Okay, I put some already out here. I kind of hope that I'm able to catch a few of those. I don't know, do I have one? Yeah, I think there's the one here. Could be. Let me see. Yep. So I'll quickly, I'm going to quickly check under the microscope whether I've got one. Yes, I've got one here. So let me quickly see, where is the scope view? Oh, it's way too much. Wrong. Here we go. Okay. Yeah, you've seen this already before. Probably. Here we go. Let's try dark field. Here it is. Yeah. It's always kind of nice, yeah, looking at these guys here. And the idea is, and that's a little bit my hope, is to, when feeding it some of the blue stained yeast cells, that we're able to see it a little bit better how the stained yeast is actually eaten up and goes through the digestive system. Okay, so this is kind of a little bit the hope that I have, and that's what I would like to try. Okay, so. So I would like to have a quick look again. Where did you get the crystal violet? You can buy it. By the way, if you are interested in buying stains, at least if you're in Europe, there is generally a problem that many companies don't like to send liquids. But there is a British company, I'm not affiliated with them. The British company is called Brunel Microscopes. And they're selling all sorts of stains in powdered form. And because it's not liquid, it should not be a problem to deliver those, right? So it's called Brunel Microscopes, and they're selling all sorts of things, yeah? So that's something that I would kind of recommend as well. So that's a little bit the thing. Let me quickly have a look at the stereo view. So I'll be kind of jumping back and forth a little bit. So far, you see actually that the crystal violet is slowly starting to diffuse into the agar. Yeah. But let's have a look if, yeah. But that's really something that I would like to see actually, if it's able to pass and move on inside. Let's go up here a little bit. That would be interesting to see if this would kind of spread. We're gonna see. I don't know if something's already happening over there or not. See this here? It's a little bit bluish over here. I don't know. We're gonna see. Yeah. I hope that we see some kind of response. Yeah. So desk view. Here we go again. So I'm going to try now the following. I'm going to try now to add a little bit of this how do you call this? Stain. However, I'm not so happy with the place of the water flea, because it's right at the edge. It's right at the edge of the slide. So you see over here, that's the edge of the slide, and it's kind of a little too close. So I'll try to move it a little bit more into the center. I'm just going to push it with a. It's better. I'm going to push it with a how do you call this? A heart is still beating. That's fine. Okay. Yeah. So what this is actually the heart here on the back. Okay. You can yeah. So I really hope that this experiment works. I'm going to now add some of this blue yeast cells. Really only a small amount. Really is, because you never know if the crystal violet doesn't also hurt. So, and all of those dots that you see, those tiny dots that you see, are actually the yeast cells. And I hope that this actually works out now, and that we're able to see. Oh yeah, we were actually able to see how the yeast cells actually are now inside. Let me go here. Yeah. Let's. Yeah, and then. Yeah. So those. That's basically. Yeah. it's actually also quite nice to see how the stained yeast cells, how they now indicate the movement of the water. And actually, what I hope to be able to see is that we slowly see that it should start to build up in here, yeah, and that we actually see the inside of the intestine start to also turn dark, yeah, because of the stained yeast. Yeah? But now we see actually it's kind of a lot of the stuff is caught in here, yeah, as well. Yeah. Yeah, they have a heart. I want those critters that they often have a so-called an open circulatory system. So this means that they do pump it, but the blood sometimes simply bathes the organs, but the blood is not circulated in a closed circulatory system like we have. Yeah, so it's kind of like the liquid inside the body is being pumped around. Yeah, yeah. So. What's this weird balloon-like organ? I mean, if you're talking about the stuff that's kind of pumping in the back, that is the heart. There are actually some experiments that you can do as well, like adding different substances like caffeine and so on. It will actually change the heart rate. Where is it now? I'm not able to see it. The heart is not quite easily visible now. It's in the back here. If you're able to see it pump quite quickly. Yeah. So that's basically simply a process of live staining. And what I also see a little bit, and that's also kind of fascinating, that the crystal violet also stains the Daphnia itself a little bit. It's not just the yeast cells, yeah, but also a little bit of the Daphnia itself. Quite a lot of questions. What are they thinking? What is he under quotation marks thinking? Most likely this is a female. Usually, Daphnia are mostly female, and they will reproduce by so-called parthenogenesis, which means that the babies will develop out of an unfertilized egg. Yeah. So the largest population usually are females. Males are only made when the conditions are turning bad. Then males are made, and then different types of eggs are made that are able to withstand dryness. But look, one of the things that I kind of like here is, and maybe I'm going to go down with we can actually see now that the Daphnia itself was already stained. Oh, it's already time for, really? Time goes quickly. Yeah, I received your questions again. Yeah, okay. So that's why in a few minutes I would like to answer those as well. So you actually, that's also something quite nice, that we see that the crystal violet also seems to be slowly staining the animal itself. What I consider a little bit fascinating and is that in previous observations, I could actually see when I was feeding something, I could actually see the food travel inside the intestine. But I see that maybe it's already full, and that's why it's not eating so much right now. It's kind of interesting here, yeah. And what's the lifespan? I don't know what the lifespan is really. How much do they eat per day? What those Daphnia, questions that I cannot answer that easily, right? Usually, I would probably, I mean, I kept the Daphnia, some of the Daphnia in there for I mean, the population as such I've kept for months. But you see there are always new ones being born and some dying off. Yeah, but the lifespan of one of them, I'm not quite sure. And I think it's quite well possible that maybe they're even able to withstand also freezing, maybe in winter. This could be. Yeah. So this would be kind of interesting. Could the stain be absorbed through its gills? This could quite well be possible, and maybe that's the reason why it's kind of bluish over here. But I actually have seen also in the past especially when you feed it some green algae or so, that they are basically also taken up, and usually they will then change the color based on the food that they eat. Yeah, so a little bit. My hope was a little bit to but there is not a lot of yeast now left, so I'll just add a little bit more maybe. But that's essentially a little bit of an advantage of this crystal violet, that it, because wow, that's way too much. Heavens. Oh my gosh. This is you know what I'm going to try it now with I'm going to add a little more water here. This might not be good for it. I cannot possibly imagine that this is let me quickly dilute it a bit again. Am I killing it? It's still going. Yeah. Yeah. Oops, okay, I guess this was a little too much for it. Yeah. Well, too much of this apparently is also not quite good. Okay, you know what I'll try? I'll try to put it back into the, maybe I'll just show you what I'm doing here. I'm going to put it back into the, here it is. Maybe it's able to recover. Okay, here we go. Yeah, but actually it's kind of interesting. You're not able to see this maybe, but actually I can actually see that it is now stained much darker than before. So let's give it another try. Yeah, let's see. I have to change the scope. Here it is. Yeah, I think it's recovered. Okay. Okay. Interesting as well. We can actually now see where the yeast can be found. Yeah. So all of those dark dots. Yeah, and I think it's now doing better again. Here on the back, you can see again the heart beating. As a matter of fact, in the book that I read, they recommended that the yeast be stained with saffronin, which is a red stain. And then those parts where the water flea, where it actually eats the yeast base, or where it accumulates, will actually turn red. Yeah, this was actually the recommendation, but we can see that it's now mostly I was kind of again hoping that it's probably going to be somewhere building up in the intestine, but maybe it's already full. So lifespan. Here's some info about the lifespan. It depends on factors such as temperature, but can be 13 to 14 months in some cold fish-free lakes. In typical conditions, the life cycle is much shorter, not usually exceeding 5 to 6 months. From Wikipedia. Thank you very much. Interesting. Yeah. Yeah. So yeah, because they're basically eaten by fish a lot. Technically, do tardigrades have an infinite lifespan if in stable conditions and perhaps no predator? It's also an interesting question, a little bit also of a philosophical question. I mean, there are many organisms that have, I mean, there are some jellyfish, what I read, that have a possibility to regenerate and renew themselves, so that they basically almost have an infinite lifespan. And also tardigrades, because they're in the so-called process of called cryptobiosis. This is kind of a when they're able to withstand extreme conditions, like they're kind of forming like almost something like cysts. then they're also able to survive extremely long time periods. So it's a little bit of also a philosophical question, I would say, yeah, on what the lifespan is, because if they're in this dormant condition, then they can survive quite long time periods as well. Yeah. Why is it not moving around? The reason why it's not moving around so much is because I did not use a lot of water. Okay. Here we can also see it. So those dots are the yeast cells again, because there is only a thin layer of water on my microscope slide, and therefore this limits the movement of the water flea, and therefore it's not able to swim away so quickly. If I were to add more water, it's going to swim around all over the place. Here it is. Jesse is already explaining this. Yeah. So so it's simply limited movement. So let's have a look, and I really hope that I don't know. I was kind of hoping to see this a little bit better. How it's Let me have a look here. But at least I can show you what's happening here already. Let me refocus this. Yep, definitely. If you're able, that's the first stage always. I put some oats on here, and you can actually see that now it's starting to slowly crawl up the side of the oat here. Yeah. So it's after maybe, I don't know, 20 minutes, half an hour, it's there's going to be it's going to be more yellow over here, because the slime mold will actually crawl up here. Yeah. So I expect this blob part here also to crawl up, either up here or to be pulled back and then crawl up here on the side. Yeah. So this is something that we should be able, now I kind of realized that there's some food here, and now we should be able to observe that. We can actually make this a little bit bigger. Yeah. And let's keep this here a little bit in view. Yeah. And how this actually works. And in the meantime, while we are observing this, I'm going to answer some of the questions here, which are also off topic. An interesting question: how do microorganisms age, and do they even age? And generally, microorganisms, they do not age, and they do not have a lifespan like we have, single-celled ones at least. So, for example, you've got a cell, paramecium, bacteria, whatever. Yeah, becomes older, becomes larger, it divides. Then you have two of them, right? Both of them are again young. Yeah, so in that sense, you could say that microorganisms have an infinite lifespan, because as soon as they become old, they divide, and then there are kind of two of them that are young, right? So it's a little bit a question of philosophy, and when we say aging, and when then this is a very human also a term, because we have some kind of a built-in limited lifespan, but microorganisms actually it works different, because they simply divide and then they're young again. So young under quotation marks. So this is so we'll be kind of observing a little bit what happens to the slime mold, and you can see, just have a look at the edge here, right? Have a look at this here. Yeah, that's important. And also what we have here, where is the scope view? Yeah, here is the water flea again. Let's see a little bit where the blue yeast starts to accumulate. Yeah. Okay, it recovered again a little bit. Yeah. So it's actually quite nice. We're able to see the nice shell of this animal now quite nicely. Yeah. So future video idea: sexual reproduction of a single-celled organisms that swap micronuclei, if possible, showing examples of live specimens doing this. Thank you for the suggestion. As a matter of fact, a few days ago I released a YouTube short showing conjugation of two paramecia. So if you go into the YouTube shorts of this channel, then there is actually one that actually shows two paramecia conjugating. And indeed, I wanted to actually make a complete video, a long-form video of that as well. And it's also a very interesting thing to look at. So what I'm going to do now is I'm going to go through some of those questions here. Okay. You know what? I'm going to, I could, I don't know, I could theoretically show you those questions here as well, but I think it's more interesting maybe to observe the water flea. Yeah. I'm going to, yeah, let's just, these are basically the questions. These are the questions that we are able to, that you're able to see in the comments as well. Okay. So I'm going to have a look. Microwaving was invented for sterilization purposes. Ah, yeah, interesting. So that is when I made the agar medium. So it was microwaving it. I hear or I read that slime molds can predict future stimuli from previous experiences. I also read somewhere that they seem to have like a very simple form of memory. Wouldn't probably call it real memory, but I suppose that there are some certain chemical signals that essentially it's somehow able to remember. So maybe, yeah. So it's able to also detect the environment and seems to show. I mean, it's not really intelligence, but some people actually say it's some kind of a slime mold intelligence because it's able to find the food and it's able to connect different pieces of food and so on. I wouldn't say it's real intelligence as such, but at least it makes the impression as if the slime mold behaves in a somewhat intelligent way. But again, one has to be a little bit careful that one does not apply human concepts to those slime molds. Yeah? But the idea is they're fascinating, right? I wonder why it's pulsating that way. So the slime molds, when you increase the speed of the video, it's pulsating, and that is because of the flow of the chemicals. There's a substance called CAMP, cyclic AMP, that's passed on through the slime mold and is used as a chemical signal. Yeah, and apparently with each wave of chemicals, that's why it's somehow pulsating. And this, I guess, is because of also of the contraction of, not the contraction, but the sliding of the actin and myosin filaments that kind of push the slime mold forward. Does the mold have many more organelles too? Yes, it does. But as we had already a look beneath the microscope, let's have a look at the scope again. Let's the other one. This is the slime mold. Yeah, you see that there are lots of things in there. Yeah, but we don't see individual cells because actually it's like one gigantic cell, so to say, yeah, containing thousands of nuclei. Those long string-like structures are parts of the filter paper, the cotton, the cellulose fibers from the filter paper. So we just ignore that. Because when I took the slime mold off, I scratched also some of the filter paper. Yeah, but you see that there is over here at least lots of stuff inside it. But yeah, it looks like a very amorphous, almost a amorphous mess. Yeah. So here is this guy again. So, that's what I wanted to show you. Now finally. Am I relieved? Okay, what is it? Look, this was actually the whole point. Do you see now that the intestine starts to turn purple? Okay, yeah, that is because of the yeast, of the blue, the stained yeast. Yeah, and we're now able to see that this purple, part here, should travel backwards, and so we're actually able to see how the food that it has eaten kind of now changes the color of the water flea. Okay, so this was actually the whole, actually the thing that I wanted to show you. Yeah, so I'm kind of relieved a little bit at least that this demonstration worked. So this is so, that's a little bit the thing. I don't have a cover glass on here, so let's be very careful. Let's do this here. Yeah, here now you see the intestine. Here, this is the heart beating. This guy is very excited, and you should not be able to follow the movement of the food. Look, it's sucked in here in front. I'm going to go down a little bit with the magnification again. Here we go. So the food is sucked in somewhere here in front. You see, occasionally you can actually see some of those cells being taken in. Yeah, and it's going to travel backwards, and then on the other end it's supposed to actually produce some waste, and occasionally you're also able to see that it kind of produces some waste here on the back. Yeah, but this purple part here should actually start to go back as it's swallowing more and more of the yeast. So at least I'm always a little bit nervous whether I'm able to show you these things that I want to show you. Yeah, but at least this kind of worked. And look here, that's where the mouth is, because occasionally we're able to see how those tiny dots are actually being sucked in here. Yeah, here were the arrow. Yeah, yeah, yep. Now again, yeah, and. Yeah, so that's kind of, and you can also see if you look very carefully, it's slowly moving backwards here as well. Yeah? So that's basically a little an example of live staining. If you just joined in, what I've done today is actually I showed you not only slime molds, but I stained some yeast cells with crystal violet, and I've been feeding those yeast cells to this water flea, to the Daphnia. Those yeast cells are those dark blue purple dots, and now we're actually looking at it as it swallows those yeast cells, and now finally we can actually see that here as well, that it's the digestive system is starting to turn purple. So maybe after a couple of minutes we're able to see this purple part move further back. Yeah, so we have to be a little bit patient, but at least this kind of can you grab another water flea that is unstained and show it side by side? Ooh, okay, yes. Yes. This is a request. I have to just reach over to get some water fleas. Yep. So that's the stained one, okay? And where I added the stained yeast. And here, and then I do want to answer some of the questions, but let's do this very quickly as well. Are you able to see this, that there are those little tiny dots floating around? Yeah. I hope that, you have to be a little bit, let's put this away. I hope I'm not spilling anything here. Let's put this away. I'll really want to quickly answer the questions as well, but yes, there's one. Excess water has to go away. There is one here. Excess water has to go away. Okay. Yeah, I think there must be two of them even. I don't know if you're able to see it. Yeah. So the request was a side-by-side comparison. Okay. So you just have seen the stained one. You want to see now the unstained one. So without, so no yeast, no nothing. So this is basically directly taken from my jar. Okay. I have to switch over to the scope view again. Where is it? I have to find, here it is. Okay. It's actually also a very nice one, yeah? And you see that here, this the unstained one, and you see that here it looks greenish, and the reason is because I've been feeding it chlorella algae, which are green. Oh, look, that's a copepod here on yeah. And so this is basically because I've been feeding it some chlorella algae. They're green, and this kind of made it look green. Huh? The intestine. Where's the? That's another one here. Okay, same story. Yeah, you see the digestive system over here, looking kind of green. Yeah, let's see if this is also taken up. Huh? And where's the copepod? I don't know. Yeah, and the stained one with the stained yeast cells is over here. Yeah, And you can see now that it's actually turning purple, the digestive system here. Yeah? So in other words, it will take on that color, whatever food it's eating. Okay, so let me quickly do the following very quickly. I quickly have a look at the stereo view here. Oh yeah, so you see that it's slowly starting to crawl up here on the side. What about this one over here? Not satisfactory. I was kind of hoping to see the blue pigment, the crystal violet, being taken up by the slime mold, but it's not doing that as quite as nicely as I hoped it would do that. Okay, so let's do the following very quickly. I just want to go through the questions. I need to wipe just a second. I got some water here that I don't want, otherwise I'm going to make everything wet. So let me go quickly through the questions. What time is it? Yeah, oh yeah. Is it possible to use methylene blue as Kuhne solution? I don't know this Kuhne solution for quick staining of bacteria. Generally, yes. I mean, methylene blue also stains DNA quite well, so you can actually use methylene blue, and I've used it to stain bacteria. Yeah. So I see no reason why it should not work. Slime mold can be found, of course, in nature, usually on wet, decomposing trees and organic material. But the slime mold that I've got here, I bought. Okay, for those of you who joined in late, this is how you can buy it. It's the dry form, and then you put it on a moist filter paper, and then it will start to come back to life. Can slime mold dehydrate and survive winter? Yes, they will do that. They are able to survive dryness. Is it possible to use methylene blue? Okay, I talked about that. What other solution may be used in order to clean cedar oil except xylol? I don't know. Cedar oil is also used, let's put it this way. I think you're referring to the cedar oil, which is used also for oil immersion microscopy, I suppose. You should not be wiping it with xylene in any case, not only because xylene is a little bit hazardous, I think that's the reason why you're asking, but because these solvents actually might attack the glue that holds the microscope lenses in place. Yeah. So generally, what I usually do is I remove if you mean the cedar oil for oil immersion microscopy, I simply use filter paper. Otherwise you can try it with alcohol. Yeah, it might not work quite as well, but yeah, it's generally I would say it is not absolutely necessary to use solvents to remove the oil. Yeah, if it's used for oil immersion microscopy. Yeah? If you carefully cleaned it with filter paper, it should be enough. Yeah, the agar dish may be more sterile than the paper one. That could be a possibility. Okay. Okay, I'm out of glass slides. Look, I'm okay talking about glass slides. Look, I'm going to show you something. Just because you're talking about glass slides, I have to carefully lift it over. This is I collect the used glass slides in here and then I wash them. Okay, so I first soak them in hot water. And of course you want to make sure that bacteria and infectious material and so on is not so much in here, but that's why I kind of soak it in hot water, and it also works. You can also put them into dishwasher, so dishwashing solution and dishwashing powder actually works quite well. Yeah, unicellular or multicellular, talked about that. They are a unique stage in between. Okay. Is it possible to stain the mold for the nucleus to see it? You know what? I wanted to try that. We can try it as well here. That's the slime mold from before. So I'll quickly add a little bit of stain here, but I would not be too optimistic about it. I tried that, but it stains so many things that essentially it might be a little bit difficult. So what I'm going to do here is I'm going to carefully lift off to make it more accessible to the stain. This is of course way too much. Okay, I'm going to put this over here, and I'm going to also use a tissue paper to remove any excess, because this is a very strong stain, obviously. And not everything is stained, so there is a little bit here in the center, so we're going to have a look at that. And then let's see if some parts are stained. Let me remove the water flea again. And let's see how this looks like. We have to make it brighter. Yeah. So that is now the stained slime mold, and yeah, I don't know if we're able to. Yeah. You see, there is so much stuff in here that actually the staining is relatively indiscriminate. It kind of stains pretty much everything that it touches. Yeah. Yeah. So what I would probably have to try, and this was just a very quick trial here, but what I have to try is I have to probably get the correct concentration, and then maybe it's possible to selectively stain some parts of the cells. But this one over here is not really very successful, I would say. Yeah. So whatever it touches, it kind of stains. Yeah. So, but it was in any case a nice try. Let's put this away again. Let's see. Where is the water flea again? Here it is. Ah, look, there is this streaming motion. Here we go. Yep, you can already see that more of the intestine now started to already become purple. Yeah. So I quickly go through the further questions here. Do you keep the plates with the slime molds in a dark place? No, I just kept them on my table. However, it does say that you should keep them preferably in a dark place. The slime molds preferred to have it darker. Yeah. So the agar is not the source of food. That is correct. In my case here, those agar plates, I did not add any yeast extract or any food. So the only food that I added are those oatmeal flakes that I put on the agar plate. Yeah? So that's kind of what's the idea. Because if I had I added some yeast extract and some nutrient, then I would have also massive bacterial growth on the agar plates, and this is something I did not want. Yeah? So the opinion on digital microscopes. This was actually a question that was asked. It depends a little bit of what you mean with digital microscopes. And if you refer to those that have a built-in camera with a built-in screen, then my opinion is that they might be quite useful if you... there's so many of them available, but I think they might be quite useful and compact if you, for example, have maybe some science exhibitions and you want people to look at them, at the specimens, and the setup should be easy and straightforward. But for my personal, as a hobbyist, I like to tinker around a little bit. I like to maybe try different cameras and connect them to computer and so on. And for me, a complete solution with everything integrated is a little bit too limiting. That's my personal view, but it might have advantages in other situations. So I would say it depends a little bit of what you want to do. I would say high-end microscope. I mean, it depends a little bit. They have got now also digital microscopes available that are quite high-end already. Yeah. So that's all. So it depends, in my view, very much on the individual device. I, for myself, have these, my setups are always like this. I have a photo tube and I put a camera on top. And I like to have the flexibility because I like to exchange cameras. I like to play around a little bit with different settings and so on. But that's a personal preference. So my opinion about digital microscopes is that it depends on what you want to do. Yeah. and if you feel, and this, by the way, this goes with every type of microscope, if you feel that you want to try it out, then try it out, and you're going to find out that sometimes it meets your expectations and sometimes not so, right? As long as ultimately the whole thing is a hobby, for many of us, and therefore as long as it brings enjoyment and enrichment of our lives, then I think it has served its purpose, right? So I personally try to go a little bit away in my philosophy from the actual technology and from the microscopes towards the observation part. Yeah, and as long as we are able to observe the things that we want to observe, we'll be fine. So, for my personal view, I like systems that allow me to play around a lot. Yeah, but this might not be in everyone's interest. Yeah, that's why these devices exist. Yeah? So, as you see, this is not a yes or no opinion that I have. It's more an opinion. It's just, what do you want? Yeah? Today I mixed myself violet and fuchsine to gram staining. Crystal violet is annoying as hell to work with in the crystal form. Everything I touch with water gets violet. Yes, honestly, the crystal violet, I show you. The crystal violet is such a strong stain. Look, that I had problems getting the stain out. You have to use alcohol to get it off. So crystal violet, if you kind of go into your bathroom and you wash it out, it's gonna stain your how do you call this, the sink as well. So crystal violet is indeed a pretty strong stain. So this is something to be aware of. I would think that slime molds would excrete their wastes outside. Yes, of course, that's what they do, is they release wastes to the environment, just like as they take up food from the environment. Yeah? The recipe is 2 grams of agar with 100 milliliters of water, tap water. In my case, microwave it for a minute until the whole agar is dissolved, and you pour the plate. Yeah. Thank you very much for this for the time lapse. I'll have a look at this. Yeah. It looks only a few regions are actually moving or growing of the slime mold. That's also correct, and I also already talked about that. The crystal violet I got, we had it in our lab where I work. But if you want to buy stains, as I already said, have a look at the webpage of Brunel Microscopes. They sell stains in powdered form, and powdered form can be sent over mail, not liquids. Yeah. The Thought Emporium has a short where the slime mold solves a maze. Yes, if you put basically food inside a maze, it's going to find its way through the maze. Yeah. I'm not aware, I did not mix any phenol. Phenol wouldn't be healthy. But I probably would guess that there is no phenol in the crystal violet, because if there were, it would kill off the organisms. Yeah, because phenol is actually toxic for organisms. Yeah, so you can also buy crystal stain from Poland, probably a bit cheaper that way. I would strongly recommend not mixing it yourself. So unless you like finding microscopic particles, yes, because it's such a strong stain and the powder is all over the place then. Yeah. What is this weird balloon-like organ in the Daphnia? I think the one that was pumping, that was the heart. Yeah. And they have, I think like other water crustaceans, they must have a so-called an open circulatory system, which means that the heart pumps the liquid within, around, and the organs are bathed in the blood, but not so inside blood vessels. Yeah. So let's have a quick look again at the stereo view, and then I'm going to have a look at some of your other comments which did not make it on the list. Yeah. Yes, now we can actually see. See, now it's starting to crawl up the food. Yeah, here on the side. Yeah. And now you also know why I made this time-lapse video, because it does take a little bit of time. Yeah. And and this is something that did not work out as I hoped, because I was kind of, but you know why? I think I know why. I was kind of hoping, I was kind of hoping that the crystal violet is going to stain the slime mold, but you see the slime mold has already retreated here. It's not here anymore. Yeah, it's up here. Yeah, so This means that I think maybe I stained a region, yeah, where there was not enough slime mold left. Yeah, yeah. So that's quite a possibility here. So we do now the following. I'm going to quickly have a look back at the scope view here at the. Let's take the arrow. Look, look. Now the whole intestine is already purple. So this means this actually could also be used as a little bit as an and yeah, up now it pooped. Did you see that? Yeah, now you can actually also use this as a little bit as you could time how long it takes the food to be digested. Yeah, yeah. So it's taken in the mouth, and now it's kind of yeah producing the wastes here. Yeah, on the other end. Yeah, but now you see that yeah, you can actually see that the whole intestine is now purple because of the stained yeast. Yeah, so I'm going to now the following. What time is? Oh, it's already 1 hour 30 minutes. I did start 5 minutes later than usual, but I'm going to now do the following. I'm quickly going to go through your comments again. Let me have a look. Yep, let me see. Most of the comments are not for me in any case. Yeah, maybe you can stain the food with mold also and dye with the yeast as you dye. Yep, but we've seen that this somehow does not. Yeah, I'd have to take a little more time because dyeing and staining the slime mold will take probably more time. Yeah. So what I might try is indeed maybe put one of those oats into the crystal violet and then see if this actually will cause the mold to also turn purple. Yeah. So Brunel stains. This gentian violet. It's that what you use tonight? No, it's called crystal violet. I don't know if it's the same thing. Yeah. So unfortunately, I don't know that. Yeah, it's crystal violet. Yeah. So no comments for me, no questions for me, no questions for me. Microwave talking because I use the microwave oven. Normally you should autoclave. The agar autoclaving is usually a pressure cooker. But I found that microwaving also works. As far as I've read, the phenol is added for gram staining and makes the dye a bit stronger. It's not necessary, but I thought with a low mid-range microscope, everything helps. Yeah, I probably would, let's put it this way, phenol is not the healthiest thing either. So if possible, I would probably try to avoid it, yeah, at home. Yeah. Do different color dyes of the food, maybe with food colors and different Daphnia, and then mix them. Yes, this is indeed what I wanted to try as well. I also want to try to get some saffron in staining. Maybe it's possible to use the red staining called the carmine staining, which is red. Yeah. And then maybe, yeah, some of them will actually work. I've also got fountain pen ink. Yeah, this is a fountain pen ink which can also be used for staining. Yeah, so I'm currently in the process of trying different things here. Yeah, Oliver, it's okay that you have been going on for 1 hour and 30 minutes. Okay, some of us were not here at the beginning. I did start a few minutes later, I have to say. Yeah, okay. What would you suggest for a stain and technique to observe cell death in fungi? Ooh, I don't know. Let's put it this way. Crystal violet and methylene blue are general purpose stains which stain a lot of things, right? But the thing is it depends a little bit what you want to say if you want to observe cell death. If you want to stain a live cell and then see what happens, then you have to make sure that this whatever stain you use does not harm the organism. And certain stains, like for example Löffler's methylene blue stain, actually contains alcohol. So it's a very convenient stain because you can directly apply it and it has the right concentration and everything. But the problem is because of the additives like the potassium hydroxide and the alcohol, it kills the cells, right? So it depends really a lot on what you want to stain inside the cell. And I just recently discovered this crystal violet, which basically has the advantage, at least the one formulation that I have, has the advantage that it does not kill the organisms, at least not at the concentrations that I'm using it. Yeah, so you see that the water flea still is kind of doing fine here. Yeah. What about regular food coloring? So basically, yes, this indeed, for example, carmine is a food color. It depends a little bit in what way the food color is sold. If in some shops they sell food coloring already mixed with sugar and which you can directly apply on your cake, that is not suitable. But if you're able to get somehow concentrated food color, then you might try that. Or for example, colors that are used for staining Easter eggs might work. You simply have to try it. And also fountain pen ink. So that's also something that you might want to try. So there are a whole range of possibilities and I would say a lot depends also on the concentration, of course. So sometimes I've seen some food colorings that were so bright that essentially they barely stained. So I have some tiny solids on the walls of my methylene blue. I assume that's normal. Maybe it could be those tiny solids. If you have, it could be maybe that these are some kind of crystals, right? If you've got some kind of a solution, you see some kind of things. It could be maybe that these could be some crystals due to the evaporation of the liquid. So this might be a possibility. So if it doesn't hurt, if it doesn't hurt in the staining, it's okay. I always say the following. You see, even if the stain is not perfectly clean or pure, as soon as you add it to the specimen, there is so much, usually on a microscope slide, so much dirt anyway on the microscope slide that it really, the small impurities in the stain bottle probably don't make a huge difference. So again, so let's have a quick, so this was basically the stained version. Some of you wanted to see an unstained version, so that's the unstained one from before. And the intestine here looks more greenish because it's been eating some algae, some green algae. And this one over here, where is it again? I've been feeding it some purple yeast cells, and now the intestine looks purple. So let's have another look, and I think I'm slowly going to call it a day for today because I've been it's definitely over 1 hour 30 minutes, and take some time for the video to kind of camera to reset. Here we go again. And what I normally would do then, I would simply film the whole thing for an hour or so, and then you can actually see how it slowly starts to crawl up the food, and then covers everything, and then it starts to look for something new again. And so that's a little bit the thing that I am going to keep on experimenting with during the next couple of days. So people, I have to call it a day today because I'm already really tired. I hope that this kind of showed you again a couple of interesting things to experiment around with. Again, as always, slightly improvised. I like doing that. Sometimes I've not tried out the experiments or the demonstrations before, so there's always a little bit of an uncertainty. But I deliberately want to keep it like that, to show it a little bit more in a realistic way. So, and what I'm going to do now as a last thing is I'm simply going to put the water fleece carefully back into the jar, because even though I've got hundreds of them in the jar, I still like to put them back. Okay, so because why not? So let's do it like this. I need my pipette here, and I'm going to simply rinse it off, and it goes in here. This goes into the other one, and this one over here with the stained yeast cells, I'm going to put in here, and we're good to go. And they continue their life happily, Yeah, but you know what I'm going to do just for the fun of it, in case you have not seen this yet, how I feed them, and then I'm really going to call it a day. Look, I'm going to feed them with here. This here is some dry yeast. Don't overfeed. And if you want to have some special diet, then what you can do is you take some scissors. This here is a tablet of algae, chlorella, and you simply scratch off a little bit of this here. So these are basically the algae that I talked about. Yeah, and now they're going to be happy for the next couple of days. Yeah, so and yeah, that's basically it. So people, I wish you all the best. I wish you have a nice week and hope that you like the live stream again. See you around next time again. Happy microbe hunting as always, and bye bye.